Welcome to everyone here and those joining us via the Zoom webinar. Thank you for joining us in this masterclass, Food Science and Technology. Now, before we begin, there are some housekeeping announcements which we'd like you to listen to. Please adhere to the safe management measures and maintain a safe distance of 1 meter apart. Sit in the designated seats here in the LT, which are marked by the lack of a sticker, and please keep your mask on throughout the masterclass. In the event of an emergency, please follow the directions of the usherers, which are myself and Anna over here in blue, and evacuate in an orderly manner. We also kindly remind everyone to please put your phone to silent mode. At the end of the masterclass, please exit the lecture theatre using the door on your left at this end and exit in an orderly manner. There will be a Q&A session at the end of the masterclass, and feel free to send in your questions via the QR code or the Poll EV link at pollev.com/Q&ALT26. That is Q&ALT26. And for those joining us via the webinar, in the event that the Zoom session ends or disconnects, please rejoin us at the same link. This masterclass will cover the fundamental knowledge of the principles of virus detection methods, including immunological methods and molecular methods, the pros and cons of each, and the application of these methods in foodborne virus detection. We will also discuss foodborne transmission possibilities of the SARS-CoV-2, as well as better risk assessments and alternative sanitization techniques that may mitigate the risk of SARS transmission in food production areas. Guiding us through this masterclass will be Dr. Li Tan from the Department of Food Science and Technology. Can we please welcome Dr. Li Tan? Hello. Okay.
All right, good day, everybody. First of all, it's a great honor of me to represent our Department of Food Science and Technology to welcome all of you to this masterclass. As you may already know, food science is actually a comprehensive knowledge, and we have quite a lot of branches. I myself was trained as a food microbiologist, or more specifically, food virologist. So in simple words, my research work is mainly to study the viruses in food. But of course, nowadays, everybody, the most familiar virus for everybody is SARS-CoV-2. Therefore, I figure out this topic, SARS-CoV-2 and foodborne viruses, so you can have a little bit connection and understand both. Right. The lecture will be divided into two parts. The first part will be talking a little bit over the virus detection methods, ART and PCR. These methods are perhaps words you hear almost every day from media, from people around. So what exactly are the principles of these two methods? And are we using the same methods in our food-related research? Yes or no? Right. The second part of the study is to answer a question whether SARS-CoV-2 is foodborne, or one step further, whether this virus can be considered as a foodborne virus or not. Okay, let's start first of all over our introduction of virus detection methods. First of all, ART. Full name of ART test is antigen rapid test. Right? If you read the word, you already notice, okay, antigen there, right? And then you may think, it must has something to do with immunology, right? Can be complicated. Good news is you actually don't need to have a lot of immunology knowledge to understand this methods. In essence, we are actually making use of this recognition between antigen and antibody. So the only thing that you need to know in order to understand the methods is antigen and antibody are two categories of molecules that they can recognize each other very specifically. It's a lock and a key, right? They have the specific structure, they recognize each other, they can bind to each other. That's the only thing you need to know, okay? Now, in order to assemble complete ART device, we will be using 3 types of antibodies, let's say. So these are information that's Okay, thanks. These are the information that you have to bear in mind for the moment in order to understand my next slides, all right? So first of all, we will have to sacrifice these very poor mice. We have to inject some protein from this virus to the body of mice, so that this mice will start to generate antibody against this virus. So in vitro, in the lab, and when we collect the serum of the mice, this antibody will recognize this protein and this virus protein and binding into each other. That's the first thing. Here we will represent this mouse antivirus antibody with this blue light blue Y. And it's because an antibody molecule looks like this letter Y. Okay, so remember this light blue Y represents mouse antivirus antibody. All right. The second category of antibody, we will need to use another animal. Doesn't need to be rabbits necessarily, can be any other animal, but has to be another type. Okay. Still, we will have to immune this second type of animal here, rabbit, with this virus-related protein. Okay, we inject this protein into rabbit, and the rabbit's body will also generate their specific antibody. Okay, it's another type. Okay, towards the same antigen, the same virus, and here we will represent it with an orange Y. Right? The third category of antibody we will be using has nothing to do with the virus. Important, huh? It has nothing to do with the virus. It is a third type of animal against the first type of animal. In this case, we call it goat's anti-mice. Okay, we take the blood from mice, we inject it into the body of the goat. So the goat's body will detect, okay, there are some alien matter, and the goat doesn't know, okay, there's some alien matter injected in my body, so I have to generate antibody. So this antibody will recognize the content from a mice, right? This is a third type of antibody, and then we represent it here with a dark blue Y. So now we are clear. We will need to use 3 types of antibody. The first and the second were from 2 different types of animals against this antigen, against our target that we would like to detect, in this case, SARS-CoV-2, right? The third type of antibody is from a third animal against the first animal. Understanding this, then we can go take a look. You're ready to go take a look how this device was assembled, right? This is a typical ART device. Perhaps you're already very familiar with. Okay, when it's ready, and we can load our sample from the very left side, and this sample zone. And then if you open it, you will actually see a pink pad here. We call it conjugate zone. It is actually fixed with some gold particles. Why this pad is pink? Why you see some pink fluids flowing over? It is from this colloidal gold particles, and these particles are pink. On top of these particles, we coat a layer of these light blue Y. Remember what is light blue Y? It's mice antivirus, right? This first type of antibody. Right, then we're done with here. Move forward this test line. You typically will see a letter T there, right? It is fixed with the second type of antibody. Remember, it was rabbit antivirus. Okay, and you may also notice there's also a C line there. It refers to the control line. It is fixed with the third type of antibody, which was anybody knows? Remember? Goat's anti-mice. Okay, this line is called a control line because it has nothing to do with the virus. It doesn't matter if you have virus or not in your sample, this line should always stay positive. Right? Why? Let's let this device work. So imagine Now you have a positive sample. That's the swab really has virus in it. And so we will load here on this pad. And here at this very end, we have these absorbent pads making sure that the liquid is flowing towards this end. Okay, it's flowing. Suppose if you really have a positive sample, this virus, when it's passing through this conjugate zone, it will recognize this blue, this light blue eyes, right? This antibody, because these antibody were against the virus. They will recognize each other, they will bind to each other, right? And then after their recognition, after their binding, they will keep on going to the test line. Okay. At this time, our virus will have a dual function. You can see my mouse. Yes, our virus will have a dual function. It's like a sandwich, right? It's like a sandwich. It's on this side, it's binding to these gold particles because of this mitigation of the first antibody. The other side is also recognizing the second type of antibody. Remember, it was rabbits against virus to also recognize this one, right? Therefore, our virus will stay as the meat of the sandwich, right? Stay inside. And that's why this pink particles will stay here. Will stay on this line to form a pink line. That's perhaps you have seen a lot, right? And whereas for this control line, the pink particles also stay here, but it's based on a different principle, based on a different reason. Okay, it's because of the recognition between these two antibodies, the light blue and the dark blue. Remember what was the dark blue? Why? They are Gold's anti-mice. Now it detects actually the presence of mice matter content. Oh, this molecule actually comes from the mice. I will recognize it. I will bind to each other. I will form into a line. So if you have a positive result, you will indeed have two lines, but it's based on different recognitions. And now you can imagine if you have a negative sample. If you don't have these green dots, you don't have the virus here, okay, still flowing on the same way, and you will see, okay, here on this line, it will not recognize each other anymore. So the particle is just a flow away without doing anything. And therefore, here on this line, it will stay empty, white. Okay, whereas when the negative sample passing through this control line, it will do the same as I showed here. So this is the principle of our ART test. Okay, finally, you got a little bit more detail over this test that we're almost doing every week. The second methods that I'm sure you're also hearing a lot recently is this PCR test. That if you have a self-test with ART and you found it positive, especially have a lot of symptoms, you will go to the doctor and normally they will do a PCR test for you. PCR, the full name is polymerase chain reaction test. Before you get to know the specific mechanism, the very important point that you have to realize is why this test is so-called more sensitive than ART. Why? It's because this methods involves a very important component, amplification. Whereas think about this ART test, it's kind of a linear detection. How many viruses you have in your sample, okay? How many viruses, they will bind to maybe one or few antibody molecules and in the end just to form the line. There's simply no amplification of your signal during the whole process. Whereas for this PCR, you do have amplification process here. So in theory, if you have one genomic copy in your starting point, as your template, right, to start with this PCR test, after 40 cycles, which is typically what we do in a lab, you will have in theory to the power 40 amplicon copies, okay? There are actually a lot of different methods in detection these DNA generated from PCR, but even if you don't know the details, it's not hard to imagine. If you have like a few thousand billion copies in this small tube, it's rather easy to detect, right? It's much more easier to detect than just a few copies from this ART test. So this is the reason, and this is the reason why PCR test is regarded much more sensitive than ART test, because it involves a very important procedure, amplification. Right. Next, I will briefly go through one type of the PCR. There are many different formats of PCR that we do, so that you have a very brief idea what a PCR is like. Okay, a typical PCR starts from a double-stranded DNA. Although the genome actually from most of the viruses we're dealing today is RNA virus, so you still have another procedure before that called reverse transcription, which is not important, as nowadays always combined with a PCR. So let's start with the real template and the real starting point of a PCR, which is a double-stranded DNA. Okay. At this stage, we need to control the temperature, first of all, to heat it up, typically up to 95 degrees Celsius, so that a DNA molecule will be denatured into two single-stranded DNA, right? Now we need to change the temperature. So a PCR reaction is actually repeatedly change a temperature profile. We change the temperature to typically around 50, 60 degrees Celsius, depending on the specific sequence of the primers. These pre-designed and synthesized primer at this temperature will anneal, bind, we recognize its complementary sequence and bind to this single-stranded DNA here. We have three small molecules prepared. Okay, we have two primers, up and downstream primers. We have also a probe, right, sitting in the middle of these two primers. The specialty of this probe, well, it looks quite similar with a primer, but it still has two buts here, right? The green one represents a fluorescence reporter, and the black one represents as a quencher. Okay, when these three components stay together as a probe, so this is actually a small segment of DNA. Okay, when this DNA is linking the reporter and the quencher, nothing can be seen. So there's no fluorescence signal at all because of the presence of this quencher. But now, if we change the temperature again, and with the help of an enzyme, okay, this elongation will start, okay, starting from the two primers that you designed to all the way going along to form into two new DNA molecules. So this is considered as one cycle, okay? So one double-stranded DNA now become two. And next cycle, we run it again, two become four. That's why if you run 40 cycles, starting from one copy, you will have to the power 40. You can quickly calculate how many in your then you will have in your tube, a lot, right? But interestingly, during this elongation procedure, the probe used to sit here will be hydrolyzed, set free into the system. Previously, they were all together. So this fluorescence was quenched. Okay, they cannot do anything. Now, if they are hydrolyzed, they are set free to the system, then this reporter is started to give fluorescence signal. So in this specific case, we simply need to collect the generation of fluorescence signal to follow up the generation of double-stranded DNA. As you can imagine, at the end of this signal, at the end of this reaction, you will have very high level of fluorescence signal. You will see, this is a positive. And which is better. You can even determine how many copies were there by the use of another other setups. So, okay, today, due to the time limit, I will not give more details anymore. This is briefly to let you have impression what these two methods are. But remember, we are still talking about food, right? Today, we are not talking about SARS-CoV-2. I just would like to start from these two methods that you're quite familiar with to get maybe interested into our foodborne virus related study. So how about foodborne virus? How about when foods are involved? Are we still doing the same, using the same methods? The answer is no. ART test, I can tell you, is definitely not sensitive enough for foodborne virus detection. Why? Because when foods are involved, things get much more complicated. On one hand, if you collect samples directly from patients, these clinical samples, typically you will have actually very large amount of virus loads from these clinical samples. That therefore, in many cases, the ART still can generate a positive samples if this patient is really shedding the virus, right? Whereas on the foods, we have so complicated food matrices, as you can see from these figures. And the usual loads of these virus particles on foods are very, very low in comparison with the clinical samples. Okay, so now a most important bottleneck or hurdle we will have to conquer before we can perform these tests I just showed you is we have to purify and concentrate these virus particles scattered here and there on the food to a very small volume, typically 0.1 to 1 milliliter in a small tube before we can go ahead with the following detection. Therefore, I can tell you a very conclusive thing is only molecular methods can be used for foodborne virus detection. And even then, the difficulty we have to conquer is much more complicated than clinical samples. Okay. Second challenge, again, when we're talking about clinical samples, we typically don't discuss about it because when you collect these samples from patients, especially if you detect large amount of viruses, you barely will doubt, oh, whether these viruses are dead or alive, because it's infecting this person. You don't need to ask this question anymore. But when you are dealing with the viruses from food, this is actually a very important question. Now, please recall a little bit of the molecular method of the PCR methods I just went through briefly. Think about it. What are we actually detecting? Are we detecting the viruses by PCR methods? No, we are not detecting virus. It's not necessary we have virus for us to have virus to generate a positive signal. What we need is actually a very small segment of this genome material. For these viruses, RNA, a very small segment of RNA. Okay. In other words, if the viruses used to be present in the food, and after this food processing treatment, after this very adverse environmental factors, the viruses were dead long time ago. What we are detecting, what we are picking up by the PCR are actually their dead bodies, even broken into pieces, okay? And then you run a PCR. Unfortunately, this small piece as your targets are still there, and you put a lot of efforts and generate a positive signal. What do you do for the moment? Okay, this is actually a very important issue because once you generate a positive signal, you have to do something, right? The stakeholder will say, oh, if we are importing a batch of foods, we do this detection, we found a positive signal of this foodborne viruses from the foods, we have to give a red light, okay? Whereas the food producers or people who want to import this batch of foods will suffer unnecessary economy loss, right? And seriously, their foods could be properly treated. It's not infectious, it's safe food. But because of a history of this virus presence, and they still suffer from the presence of these very small segments of RNA, and then they get rejected. So we lose a lot of money, we waste a lot of food. Okay, so now the question is, what do we do? And my job or my research is actually to deal with these challenges and to develop new methods for foodborne virus detection. And you are definitely welcome to join me in the future. All right, so let's come to the second topic of today. Is thus COVID to foodborne. This is actually a question I was constantly asked from the very beginning of this pandemic. People ask me, okay, is it possible that people will get infected from this virus? Or more straightforwardly, can we regard this virus also as foodborne virus or not? Before we start, first a little bit basic knowledge over foodborne virus. I've been talking about foodborne virus, but maybe you are wondering already in your mind, what viruses are we talking about? And which virus is actually regarded as foodborne viruses? There are quite a few. Generally, we can divide foodborne viruses into two categories. There's one category of viruses causing gastroenteritis, represented by norovirus. Perhaps now and then you hear this word from the media. The second category is more famous, I would say, is foodborne viruses causing hepatitis, represented by hepatitis A virus. And this virus is much more widely known because it's causing a lot more severe symptoms. Okay. And it's also quite prevalent. You think, okay, now I've barely heard about it, but actually it's still quite prevalent. Taking the example of human norovirus, yearly is causing almost 700 million cases globally, right? It's also causing many deaths, especially for immunocompromised people. That's the same actually for SARS-CoV-2. Most of the known foodborne viruses are not zoonotic, meaning if you get infected, for sure you are getting actually a trace back the source is actually from another infected person. Okay, although the human excretion, stool and vomits can contaminate a lot of components, in the end, contaminate directly to the person or via the environment or via the drinking water or the foods. The foods are quite commonly, quite often involved in this transmission. So how about SARS-CoV-2? These are the news I collected actually triggering this question, I would say. Therefore, we quickly looked up a lot of literature and we published this opinion, this literature review article. So what makes a foodborne virus? So what kind of conditions these viruses has to fulfill to be regarded as a foodborne virus? And by comparing with coronaviruses, so SARS-CoV-2, with human noroviruses. Let me briefly introduce you our conclusion, as the time limit is almost there. The first one is, we believe if a virus would like to be so successful as a foodborne virus, first of all, it has to conform some conditions. The first one, we will have to make sure that the patients really get sick from the viruses contaminated in the foods they consume. Okay, that's a very important condition. Okay, and currently I haven't been reading any solid epidemiology evidence showing any of these patients really get sick from the foods they eat. There's no epidemiology evidence. Well, which is hard to justify, right? But still, there's no evidence. Secondly, a foodborne virus is a virus typically follow, we call it fecal-oral routes, meaning this virus would actually cause infection in our digestive tracts, whereas SARS-CoV-2 mainly cause infection in our respiration tract, right? And these are some example data showing how unlikely they are causing infection in our digestive tracts. Okay, the purple bars shows rotavirus, which is another enteric viruses, adapt very well in our intestine. Whereas you see for SARS-CoV-2, the red bars are very low in comparison with rotavirus. So in our stomach, there's already number one hurdle. They don't survive in low pH. They die off very rapidly in our stomach. Whereas if we go to the small intestine, yes, indeed, they can replicate in our small intestine, demonstrated by this in vitro cell culture model. Luckily, we still have another hurdle, which is our large intestine, that we will see, okay, these viruses can replicate in the small intestine, but when they enter into the large intestine, quite likely they are inactivated again. So in the end, if you detect viruses from the fecal samples of the patient, quite likely they are already inactive. Okay. So that's the first aspect. The second one, again, it's not that easy actually to survive as a foodborne virus in our environment and now on the food, because our food chain had introduced a lot of adverse conditions for these viruses to survive. So they have to be very, very tough, right, in the environment. Therefore, in order to justify whether they are successful or not, we have to first show detection or screening results of these viruses indeed from the food, which we have, like the pieces of news I show you, but most of these results were generated by PCR methods. And I already told you, how can you make interpretation of PCR methods? You found this virus, does it mean the virus is really infectious? Why not, right? Secondly, they have to survive very well on the surface, in the environment, and on the food. And you may say, yes, I read from the news, and they can survive on the surface for 2 days. They can survive on the food for 4 days, as this evidence I found also. But you must see this comparison. If you read in the literature, see how tough a foodborne virus can be, and you will very quickly make your conclusion, because in order to evaluate the survivability of foodborne viruses on the food or environment, normally we are measuring like in weeks or months. And so they easily survive very well on foods for such a long time, a lot longer than SARS-CoV-2. Also towards these inactivating strategies, nowadays everybody's using this alcohol-based sanitizer, right, to eliminate SARS-CoV-2, these viruses from your hands. A very simple conclusion I can share with you is ethanol, so alcohol-based sanitizer, does nothing to foodborne virus. So they stay fully infectious in ethanol. They are so tough, right? So all these evidences in total supports a conclusion agreeing with CDC and ICMSF. A SARS-CoV-2 contamination on the food is definitely a hazard. Nobody likes this virus to contaminate on their food, but it's not equal. It doesn't equal with a risk. Okay, when we are trying to set up the priorities in controlling and the spread of these viruses, foodborne transmission should definitely not be set up as a priority, right? Because there's no clear evidence yet how important it is. Now you may ask, then you say, okay, it has nothing to do with food, and why you are still talking about it? No, it has something to do with food, but in another sense. If you try to search a little bit, you will see another clusters of SARS-CoV-2 outbreak related with food processing, which is mainly these meat processing plant and abattoirs. And these news were already from back to 2020, but in a very recent Hong Kong outbreak, I noticed it's also again from these abattoirs that they have a lot of outbreaks and they barely have any meats, fresh meat to eat anymore. What's the reason? It really depends on the working environment of these workers. Typically in these food processing plants, they stand very close to each other, and so very high density of this of these workers. They work in low temperature and high humidity environments, which is very much in favor of the virus survival. And also their processing plant normally is equipped with this aggressive air movement and very poor ventilation system. So if one worker is infected, it's quite fast spreading everywhere and to infect other workers. And lastly, maybe it's hard to imagine, but that's true. These environments are typically very noisy. And so the workers have to shout to each other, generating a lot of aerosols. That's all the factors that we believe why a lot of outbreaks can happen in these environments. And therefore, for our food industry to consider is maybe rearrangement of the sanitation problem or the air sanitation, and to protect the workers. And one step further, to prevent food security crisis, right? To make sure we still have people working in the food industry, we still get food, right? That's the real thing. All right, so here I come to the summary of my lecture. Hopefully you get interested in foods in foodborne viruses, which still remain as a great challenge. Yeah, so I will stop my lecture here.
Thank you, Dr. Lee Tan, for guiding us through this masterclass. We will now be opening this floor to questions. You may send in your questions by scanning the QR code, which will be up shortly, or via the link, which is polev.com Q&A LT26. And if you are rushing anywhere, do feel free to leave anytime. But for those who are interested in the Q&As, do stay back and you can listen. Okay, now goes.
The answer is simple. They don't like the environment in the big intestine. Yeah, we have a lot of molecules like enzyme and so on, pH, which may inactivate the virus quickly. Yeah. How this is a food science course? Is it possible to apply the skills you learn in the course? Yeah, definitely. I think this is a good question. Food science and technology is really highly interdisciplinary. I myself, I'm currently working in collaboration with scientists from everywhere. Biology, chemistry, yeah, statistician. Yeah, it's really highly. What are the career prospects of this course? Dr. Liang would like to answer this question.
Okay, so career prospect, actually, I can't answer in 1 minute, but if you apply and you're offered a position, a place in food science and technology, I actually have a session, after you're offered FST. I have an engagement session with those who have been offered. What are the career possibilities? But in short, if you really want me to answer, there are, you can work in any food-related industry, but even pharma. So those are the two most common ones that our graduate work in. So anything related to food, which means government organizations, private food manufacturer, food ingredients company, as a flavorist, food product development, regulation, health promotion, nutrition, all these are just examples, okay? But there are so many things that a food science can do, even in the media, as a reporter, a journalist, you can also work as that if that is one of your interests, right? So in short, that is what I have for you. Next one.
Which I was told there's only 1 or 2 questions allowed.
Oh, really?
Quite tight schedule. I don't know.
Hi, so can those on the website or on the webinar, please vote for 1 or 2 questions, after which the remaining questions, please redirect them to the email we'll be sharing with you because we're short on time. So we'll wait for a minute and answer the question with the highest vote. Thank you.
If I don't meet the IGP, can I still come into FST? I am still interested. Okay. So if you don't meet the grade point average, okay, whatever that scores is that you calculate to come in, basically for food science and technology, you have to apply direct admission. Okay, that means when you fill up the form, you have to choose food science and technology as your major. But if you can't get in that route, because that route is the one that you must meet the cutoff point, but the cutoff point is not the same as last year, yeah? So don't ask me what is the cutoff point because I don't know, all right? So, but if you can't get in that way, you can still come in via CHS. That means you apply into CHS. We do take in a certain percentage for those who get into CHS to come into food science and technology. Okay. What overseas opportunities are there for this course? Oh, the overseas opportunities, too many. Okay. Actually, in our FAQ, we also cover that, but definitely in terms of industrial attachment, it can be overseas. In terms of exchange program, just like any other students in the university, you can go for exchange program. A lot of our students also apply for summer programs. Some go for NUS Overseas College, NOC, if you have heard of that. If not, search the web, NOC. A few of our students also go for that. So majority go for exchange program and industrial attachment overseas, okay, in terms of exposure as well as summer program. So those three things are the main ones. What is the teaching style like for this course? Okay, how often are lab sessions conducted? Any wet science, which includes food science and technology, Department of Biological Sciences, chemistry, they all roughly have around the same number of lab sessions, okay? But the way we organize the lab sessions may be a little bit different from one department to another department. But in totality, you get exposure to wet lab, definitely, okay? Now, the thing is, how are the teaching styles like? I'm sure Dr. Li Tan's style and my style are very different. And even my style, when you're in first year, second year, third year, fourth year is different. Okay, so I teach every year, year 1, year 2, year 3, year 4. And my style for every different year is totally different. You can ask existing students, they will tell you that. Do you think there is future for food science in SG? Oh, well, you just have to ask question. Do you eat? If you continue eating, there will be future for food science. How can I prepare myself better if I would like to, if I would like to ABA into food science? What does that mean? Oh, you mean the discretionary admission? How do you prepare yourself? If you have passion in food science, we will see it. So if you are getting in through the interviews, we look for people who are passionate about the subject. You really have interest. We don't just look at exam scores. Exam scores is not something we look at, okay? But of course, when you apply into university, there is a cutoff point based on your examination results because we have to draw the line somewhere. So that's an easy way to screen out people. But in reality, what we really want to recruit in any department, if you ask any lecturers, is the people who are really passionate about the subjects, not so much about whether you get straight A's or not. How does one enter food science as a minor or second major? Okay, so those people who wanted to do second major at the moment, because the spaces are limited, we need lab spaces. Therefore, you must major in chemistry. Your first primary major must be chemistry. Then you can do a second major in food science. But if you do a minor, it will not be food science. It will be nutrition only. We do have second major in nutrition as well, okay? So second major in nutrition is free for all. Anybody who sign up for it, basically you can get it, but it's still classified as restricted because we still have lab sessions. We see whether it explodes or not. If it doesn't explode, we will take you in general. But for the food science second major is only for chemistry primary major.